Welcome everyone to today's LSTM seminar. It's my pleasure to introduce Professor Kathryn from Manchester University. And just a bit of background on Kathryn. Kathryn originally did a PhD, I hope she doesn't mind me saying, back in the 1980s in the University of Nottingham, in the lab of Derek Wakelin. And I think it's fair to say Kathryn's one of the pioneers of the Trichuris muris mouse model, because at that time I think it'd just been established.
Yeah.
And Kathryn did some of the very early studies looking at the genetic basis of resistance in different mouse strains to whipworm infection. And then afterwards moved on to Manchester University and was very successful in getting a senior clinical research fellowship from the Wellcome Trust for a period of over 10 years at Manchester, and then being promoted to chair in 2009. So Kathryn's current research focuses are on the mechanisms by which gutworm protective immunity is mediated and how antigen-driven inflammatory damage is caused by infection is resolved. And these interests encompass macrophage biology, immunology of wild mice, and also vaccine development. And I have to also mention that beyond immunology, Kathryn's also latterly been applying the mouse model of whipworm infection as a preclinical screen to also test efficacy of novel anthelmintics against Trichuris. The title of Kathryn's talk today is Understanding the Drivers of Chronic Infections, Circadian Rhythms and B Cells. Thank you, Kathryn.
Thank you. Thanks very much. It's always a pleasure to come to Liverpool, so I was really delighted to receive the invite from Joe. And thank you also for those really kind words. It's always interesting to hear your sort of life history relayed. So as Joe said, I've got a long-standing interest in soil-transmitted nematodes. And I think many of you in the audience share that interest, maybe in different types of nematodes. I know many of you are interested in the tissue-dwelling nematodes. And it doesn't really matter what your sort of favourite parasite pathogen is. Many of them have developed lifestyles that are chronic, they're persistent, they're long-lasting. And there are lots of things, factors that could underwrite that longevity. And they will come together and make an effect on the ability of that parasite to survive. But what I wanted to share with you today is some of the work that we've been doing on perhaps less well understood drivers of chronicity. And that is the circadian rhythms. And then at the end, I'll show you a little bit of data on B cells. So what I'm going to do, I'm going to spend a few minutes setting the scene and introducing you to the soil-transmitted nematode that I've been working on for over 25 years, and that's the whipworm or Trichuris. Then I'm going to spend a bit of time talking about circadian rhythms, how circadian rhythms affect the outcome of infection with whipworm, and then right at the end I've put a few slides in to show you some of our unpublished data on B cells. So that's the plan of attack. So, of course, as many of you know, soil-transmitted helminths, they're extraordinarily prevalent. So they're estimated to infect over 1 billion people, and these are chronic. They're chronic infections of man. And I've always found it quite shocking that many of these diseases are, of course, entirely preventable if you could get financial investment to improve sanitation, education, deliver drugs and develop vaccines. So probably a four-pronged attack. And certainly in the context of vaccine development, we require a better understanding of immune responses to infection. Oops. So soil-transmitted helminths include whipworm. This is the human whipworm. Let's see if we can get this to play. So this is an endoscopy up someone's backside, and it's got the little camera on the tip, and you can see as this endoscopy snakes its way up the large intestine, the mucosa of the gut is really literally littered with whipworms. These little white snakes on the surface of the gut mucosa is a whipworm infection. So these infections can have a massive effect on a child's life, and that's shown very nicely here on this sort of health education poster from India, in fact. And so worms will reduce children's ability to attend school, they do less well at school, adults have reduced worker productivity, and that impacts on the economic prospects of that country. And of course they cause malnutrition. So massive impact on people who live in areas where these worms are endemic. Now over on this side, as a way of introduction, I've got the mouse species of whipworm. So this is Trichuris murus, this is Trichuris trichura, and this is a little video of Trichuris murus in the gut of a mouse. So now if I introduce you to Trichuris murus, so this is a cecal-dwelling nematode of mice. We use it in a number of ways. So we use it as a tool to probe the immune system, but also, most importantly, it's a model of human trichuriasis caused by Trichuris trichura. It's a very good model. We know that it's genetically very similar, it's antigenically similar, it's physiologically similar, it drives a similar sort of immune response in the affected host. It's a fascinating parasite to work from the point of view of just parasite biology. It's partly intracellular, which is an extraordinary niche for what is a 2 centimetre long parasite when it's an adult. So there's some nice images here on the bottom. This is a parasite that's been removed from the gut. This is the back end, and this is the long, thin front end called the stichosome. You can see how it gets its name, whipworm. And this front end, the stichosome, is the part that embeds within the gut epithelial cells. So you can see here this sort of snake-like tunnel on the surface of the gut mucosa. That's the tunnel. So if I was to take a transverse section from here to here, this is what you'd see. So these are the epithelial cells lining the gut, the enterocytes, and here's the head of the parasite. And it seems to form a syncytium within which it lives. Again, a nice histology slide here. This is probably just one worm. This is the back end of the worm, which is loose in the lumen. And then you've got the head of the parasite, which is threading its way through the epithelial cells. So many of you will be familiar with this life cycle, but I just want to showcase some of the images so you can get a really good feel for what this parasite is like. This is the adult parasite. It will shed eggs. It becomes patent 32 days post-infection. The eggs come out in the feces. When mice ingest the eggs, embryonated eggs, then the L1, the first stage larva, will hatch. And these larvae invade the epithelial cells in the cecum, and there they develop. They molt from the L1 to L2, L2 to L3, L3 to L4, and back to adult. So this is an X-ray tomography image of the adult parasite, which I think it really does give you a splendid feel for what this parasite is like. So in this instance, we've partially removed the parasite from its tunnel, so we've pulled it out of here a little bit. And as this spins round, you can see a beautiful detail. It's a beautiful parasite, lovely detail of the stichosome, and indeed you can probably make out the individual cells which make up the stichosome, the stichocytes. This is a male parasite, so when it spins round again, you'll be able to see the copulatory spicule, which is going to appear here, just coming in here. So it really is a fascinating parasite. We're beginning to understand some aspects of parasite biology. It holds still a lot of secrets. One of the things that we do know is the trigger for egg hatching. So this is work that came out of my colleague's lab, Richard Grens's lab, a few years ago. And we know that the trigger for hatching is actually the bacteria in the cecum when the eggs arrive into the large intestine. So if you keep your eye on that middle egg, you'll be able to see the L1 hatch. So it's become active within the egg, and then it's going to come out of this operculum here. Once it's out, it goes straight down, burrows down to the crypts, into the cells lining the crypts, and starts its molting cycle. Oops. So that's the parasite. So again, for many years, we've known that one of the powers of the model system is that different inbred strains vary in their ability to expel the parasite. So this is percentage infection here, and this is days post-infection here. If you're going to get rid of your parasites, you need a type 2 response. And you can see that some strains of mice expel the parasite quite quickly, some take a little longer to do so, and then 1 or 2 mouse strains are completely susceptible to infection. So you need a type 2 response to get rid of the parasite. So the question we ask, the burning question if you like, is what influences the development of a type 2 response? And again, there are lots of things that can do this. Host genetics plays a role. Level of infection plays a role. And I'll show you one slide each on these. But most recently, we've found a role for circadian rhythms or time of day on what happens to the worm within the host. And so I want to share that data with you this afternoon. And then, if there's time, I'll show you a little bit of data on B cells and how they impact on the way the type 2 response develops in vivo. So host genetics, really simple slide, 2 different strains of mouse. This is the AKR mouse, this is the C57 black 6 mouse. If you give these mice a high dose infection, so typically 150 to 200 eggs, an AKR mouse will make a type 1 response. So you can see that IL-4 levels are not different to naive levels, nor IL-13 levels, both type 2 cytokines, but the gamma interferon cytokine is significantly elevated. What happens to the worm? It survives, it persists, it's chronic. If you do the same experiment but in a black 6 mouse, now you can see that in addition to the gamma interferon, we've got a significant upregulation of these type 2 cytokines, and now the worm is expelled. So host genetics is important. We also know that level of infection is important. So in this instance, I've got the same strain of mouse. They're both C57 black 6 mice. And this is the experiment we've just looked at. So this is a high-dose infection. The worms are expelled, and we get a mixed Th1, Th2 response. But look what happens if you give a low-dose infection. So now we're talking 10, 20 eggs. Now you're getting persisting infection. You've lost your type 2 response, and you've got upregulation of a type 1 response. So level of infection is very important. So what about circadian rhythms? So this schematic is just to remind you that if we plot worm burden with days post-infection, if the mouse is going to expel its parasites, it's going to make a Th2 response. If it doesn't expel its parasites, it's going to make a Th1 response. So these 2 guts are taken from the same inbred strain of mouse. They're both from C57 black 6 mice. Both groups of mice were given the same level of infection. The only difference between the gut from this mouse and the gut from this mouse was the time of day that we gave the infection. So this mouse had its infection at 7 a.m. and this mouse had its infection at 7 p.m. So that was our first clue that time of day or circadian rhythms were going to be important in chronic infection. So what are circadian rhythms? So it's like an internal body timer that allows us to adapt our physiology to changing environmental cues. We live in an oscillating environment. And the way it works is that light comes in through the eyes and it hits the so-called suprachiasmatic nucleus. This is in the hypothalamus. It's called the master clock of the body. This is a little histology slide which shows you the suprachiasmatic nucleus, and one of the key clock genes, period 2, has been stained in green, so you can see period 2 is localizing here. So light comes in, it hits the master clock, and then the master clock entrains the peripheral tissues. And it does this via nervous and humoral systems, we think, and that's still very much work that's ongoing with bona fide circadian biologists. We also know that other environmental cues can entrain clocks, including food. So hold that in your head, because that will become important later on. So these local peripheral clocks, they're critical to how our tissues work, and you find clocks in almost all body tissues. In fact, if you looked at the transcriptome, around 1/4% of the transcriptome is under direct circadian control. So really, really important. Endogenous clocks control many different types of physiological rhythms. So I've put a few on here. Our sleep-wake cycle, inflammation, body temperature, feeding, metabolism, they're all, we all see circadian rhythms in those aspects of our physiology. When those clocks go wrong, we get disease. So you see disruption of circadian rhythms as we age. You can see genetic mutations, you see disruptions in disease states. Also lifestyle can lead to disruption of your circadian rhythms, and shift work. This sort of arrhythmia that is shown here is a common hallmark of many cancers and also chronic inflammatory states. So having a good circadian rhythm is important to our health and well-being. At a sort of molecular level, we know well how that works, and of course the Nobel Prize was awarded to 3 circadian biologists at the end of last year. So I've put a very simple version of the molecular clockwork on here. So clock, BMAL, period and cryptochrome are clock proteins. And what you see is if you get transcription of clock and BMAL, and these form a dimer, and they activate the transcription of 2 more clock genes, period and cryptochrome. Period and cryptochrome form a repressor complex, and they feed back and turn off the transcription of clock and BMAL. Because this transcription is now turned off, the amount of the repressor protein gradually degrades, and eventually clock and BMAL are no longer repressed, and that transcription begins again. So that's a very simple view of this negative feedback loop. There are also additional loops going on here as well, but these are the main players, I think, in the molecular clock. So what, of course, that means, immune systems have a molecular clock, and although we don't know the exact molecular mechanism that links the clockwork to an immune output, we know that it's very important. And what it means in terms of what we're talking about today is that an immune cell that sees parasite antigen in the morning, might see it very differently to how it sees that molecule in the evening. So I've got a very simple example of this on here. So this is just macrophage responses to LPS. And they're all showing the same thing, so we can focus just on the IL-6 output. So they're macrophages, they're in vitro, they're shown LPS at 2 different circadian times, either CT0 or CT12. And you can see if they see the LPS at CT12, they make a significantly greater IL-6 response. And the same is true across most of these cytokines, but not TNF. This is protein, this is mRNA, same thing. So that's a very simple sort of single cell, very trimmed down, a cell type and a single stimulus, what happens, and there's a clear circadian rhythm. So the question we wanted to ask then is what happens to the outcome infection if mice are infected at different times of day. So we used what we call ZT0. So this is ZT, it's a time giver. It's a way of describing any environmental cue. In this context, it's light. So ZT0 is lights on, or 7 a.m. ZT12 is lights off, or 7 p.m. So some of these experiments were really fun to do. They were done in pitch black, wearing these night vision goggles. So you're doing your oral gavage in the dark with your night vision goggles on. So it was an interesting experience. Let me show you some data. So the mice were infected at day 0, and then we followed the infection through post-infection. This shows you the worm burden data, and here are two groups, ZT0 and ZT12. In trichuris infection, the parasite isn't expelled before 14 days post-infection. So this shows you that all mice got the same level of infection, and that was monitored at day 13 post-infection. But if you follow that out, at day 21, you can see the 7 p.m. infected mice are significantly more susceptible now than our ZT0 infected mice. This is just a delay in expulsion. It's not a complete switch to a susceptible phenotype. By day 28, the worms are going in both groups. But it looks like if we infect mice at ZT12 or 7 p.m., we see a delayed ability to expel the parasite. So of course we looked at the immunology, and our type 2 response is shown here. So ZT0, we're getting a significantly greater G1 response, significantly elevated IL-13 response compared to our 7 p.m. infected mice. And if we look at the Th1 parameters, now you can see that we've got an increase in Th1 responses evidenced by this high level of IgG2A and high levels of gamma interferon. So a Th1 response, which is associated with chronic infection. So we then wanted to confirm that in a way that didn't involve light reversal. And we used food entrainment to sort of flip the intestinal clock. So in this instance, we used, it's called a reversed feeding regime. So along here we've got, this is just meant to be from sort of day 1 through to when we culled the mice. We've got our day in white and our night in black. And we've got night-fed mice, so these were fed 6 hours. For 6 hours, food was made available for 6 hours in the middle of the night, or we made the food available for them for 6 hours during the day. So in effect, mice normally feed at night, so in effect we're forcing mice to feed during the day. So we put them on this restricted feeding regime for 2 weeks. We then infected them, and both groups of mice had a ZT0 infection. We left them on that reversed feeding regime for a further 7 days, so 7 days post-infection, and then they went back onto ad libitum feeding until we culled them. So this data just shows you the success of that reversed feeding regime. So we're looking here at 3 different clock genes. We can just, they all show the same sort of thing, so if we just focus on the period 2 output. In our night-fed mice, you would expect period 2 to be more highly expressed at ZT12 than ZT0, which is shown here. But if you day feed your mice, it's flipped, it's reversed. You've now got more period 2 being expressed at ZT0 versus ZT12. So that just simply says the reverse feeding regime has flipped the intestinal clock. So what happens to our worms? And so what you can see here is that our night-fed mice infected at ZT0 are expelling their parasites, but the day-fed mice are not. So we've altered their ability to expel the parasite by simply altering their feeding regime, which has had an effect on the local intestinal clock. If we look at our Th1 and Th2 parameters, this was, to be honest, this was a bit of a surprise to us. We found both upregulation of both Th1 and Th2 markers in those day-fed mice. So you can see that G1 is up, IL-13 is up, but so is IgG2A and gamma interferon. So what we think is going on here is that we've got a desynchrony between the central clock after the ZT0 infection and the local peripheral clocks, such that you've got a type 2 response in addition to your type 1 response, but that type 2 response can't coordinate the local effector cells, whatever they might be, because they're at a different phase of their circadian cycle. So then we embarked on the very long process of trying to work out mechanism. One of the things we did was to start off looking at the leukocytes in the draining lymph node at the time of T-cell polarization. So this just shows you our gating strategy. So we looked at neutrophils based on Ly6G. We picked out our B cells, CD19, B220 positive. From the CD19 negative, B220 negative, we then took this down to stain for NK T cells and T cells. We separated our T cells into CD4 and CD8. And then for our surface negative cells here, we looked at dendritic cells. So here we've got CD11C positive, MHC class 2 positive, conventional DCs, which we then broke up into different subsets based on CD11B and CD103 expression. So could we see any differences in those subsets? And the answer was yes. So there was no difference in the total cellularity in the mesenteric lymph node, which drains the site of infection. T cells in the day-fed mice were significantly higher. Conventional DCs, i.e. total conventional DCs, were not significantly different. But when we looked at those DC subsets, we saw some very striking differences. So CD11B positive, CD11B high, CD103 negative DCs were higher in our day-fed mice, as were the CD11B positive, CD103 positive cells. So that was really interesting, and it gave us our first clue, if you like, that dendritic cells were going to be important. So one of our big worries, or the circadian biologists were worrying about this in the context of these experiments, was that infection can just disrupt circadian rhythms, and so you've got a sort of systemic effect. So we did some whole body telemetry just to check that rhythms were intact. So we looked at temperature oscillations and we looked at activity. And so the most important point from this slide, so this is a ZT0 infection, this is a ZT12 infection, this is body temperature. So we implanted these probes into the peritoneal cavity, and then we monitored them throughout the course of infection. We turned the probes on before infection just for 6 days, so they recorded for 6 days. They recorded for 6 days early post-infection, and then the last 6 days of infection. So that's how we did the experiment. But the most important point is that infection doesn't stop you having a clear circadian rhythm in body temperature. So the main thing is infection doesn't just make a mess of your circadian rhythms. The second most important point is that time of infection doesn't matter. You've got the same pattern of rhythmicity in body temperature. What you can take from this is that infection significantly alters the amplitude of your temperature. So post-infection, in both groups, you get an increase in body temperature, and this is consistent with a decrease in the basal temperature, which is shown here and here. So not only were mice rhythmic in terms of their body temperatures, but also their daily activity patterns were also independent of infection and independent of time of infection. So this is shown here in terms of activity over a time series. And you can see that if you're infected, you have a significant reduction in your daytime activity, whether you get your infection at 7 a.m. or 7 p.m. The interesting thing, again, the clock biologists are really interested in this, is that what happens in an uninfected mouse is that you get this little anticipatory burst of activity before lights off. And our infected mice have lost that anticipatory behaviour. And what that means, or whether we follow it up, we'll have to wait and see. So we having satisfied ourselves that infection wasn't just completely disrupting circadian rhythms per se, we then went back to our hypothesis that it was a dendritic cell clock that was important and underpinning the difference in susceptibility to infection. This was evidenced by those DC subsets that we'd looked at, and also the fact that the food reversal study was only maintained for 7 days post-infection. So it's something very early that's underpinning the final output based on the Th1-Th2 balance. So we made a clockless dendritic cell mouse. So this is the core clock gene BMAL, which was floxed and then crossed with the CD11C Cre driver. So CD11C Cre BMAL floxed mice have dendritic cells that don't have BMAL. These mice also have a period 2 luminescence in there as well. It's a reporter gene. So we can track their rhythmicity just using a bioluminescent readout in vitro. So these are bone marrow-derived DCs. This is a wild-type trace, which is just what we'd expect, nice and rhythmic. We get the peaks and we get the troughs. But when we take the clock out of the dendritic cells, then they really become arrhythmic. So we were satisfied that the clock gene had been removed from the dendritic cells. So then we looked at what happens post-infection. So here's worm burden. Here's our BMAL floxed mouse, which is now in effect our wild-type mouse. It's not been crossed with the Cre driver. And you can see, just as before, our mice infected at ZT0 have a reduced worm burden compared to the mice infected at ZT12. So that's just as we'd seen before. But you lose that type 1 phenotype in the ZT12 infected mice. So these two groups are not significantly different, and that's the key. So we think that BMAL may be a really important component in that Th1 driving response that we see at ZT12. And if you look at some of the Th1, Th2 parameters, that sort of fits with that hypothesis. So you can see that IgG1 is down in the ZT12 infected BMAL floxed mouse, which fits with the increase in worm burden. But we don't see a difference between these two groups. So we thought the dendritic cell looked like it was a key component of the circadian regulated phenotype that we were seeing. And so we wanted to drill more into mechanism. And the way we decided to do that was to use bone marrow-derived dendritic cells as a surrogate, if you like, of the tissue dendritic cell. So this is a similar trace to the one I've shown you before. And here are wild-type cells, nice and rhythmic, and these are our clockless dendritic cells. And we took bone marrow-derived dendritic cells across a long time course in vitro. They were synchronized to start off with, and then they were cultured for 48 plus hours. And we exposed them to parasite antigen. So we showed them ES, excretory secretory products, at either the peak or the trough of their circadian cycle. We harvested those cells 3 hours later because we wanted to do RNA-seq on those dendritic cells. So this is the first set of data is just quantitative, and this shows you, this looks at clock-regulated genes in unstimulated cells, so cells that hadn't been given a parasite antigen. And this shows you the number of genes that vary in an unstimulated wild-type dendritic cell between its peak and its trough. So what's different in gene expression here versus here? And 70 genes came up as by being diurnally regulated. Thankfully, in our clockless dendritic cells, only 4 genes showed any difference. When we exposed the dendritic cells to parasite antigen, then the number of diurnal genes that were stimulated increased, which is sort of what you'd expect. So we've got nearly 600 genes now in our wild-type dendritic cells that are differing between peak and trough, and certainly more in our clockless dendritic cells, but a lot less than you see in the wild-type dendritic cells. So then we went on to do the RNA-seq to look more qualitatively at what the genes were that were being regulated. So the first analysis that I'll show you is a difference between the wild-type dendritic cells at peak, so here, versus trough, over here. And when we looked at those genes, we analysed the top 40 genes using string analysis, if any of you have come across that. And we found that there were significant differences between the wild-type DCs between peak and trough. So these are genes that are down-regulated at peak. And in our interconnected network that we saw by string analysis, IL-12 beta was sitting at the heart. And if we do pathway analyses on these genes, the significant genes that are down-regulated at peak versus trough, then you find you've got some type 1 correlated pathways. So IL-12 mediated signaling, IL-23, IL-27 are all significantly down-regulated at peak versus trough. The next analysis we did was to compare our wild-type dendritic cells and our clockless dendritic cells at trough. We did the analysis at trough because we thought, so this is just a heat map, but even by eye, you can see at peak there's not as dramatic differences as you see at trough. So we're comparing wild-type and clockless DCs at trough. And I'll show you the data a different way this time. This is tabulated. So this is looking at the top 20 genes, which are significantly down-regulated in our clockless dendritic cell versus our wild-type dendritic cell. And what you notice is that you've got an enrichment in genes associated with driving type 1 responses. So here, IRF7, we've got IL-12 beta here as well, and some other genes as well, which are linked with type 1 mediated immune responses. If you look at the pathway analyses, interestingly, the same pathways now pop out again. So you've got IL-12 signaling is significantly down-regulated in the clockless dendritic cells, IL-27 mediated events. I mean, interestingly, we've known for years that IL-12, if you deplete IL-12, your mouse will become resistant to trichuris, and IL-27 knockout mice are resistant to trichuris. So that data has just been published in scientific reports. It came out at the start of this year. And this cartoon is just to try and sum up what we think is going on here. So if you get eggs coming into the gut, these will hatch and they'll grow into worms. If you're infected at ZT0, then you'll get a type 2 polarized immune response. You get a G1 and IL-13 as your type 2 signatures, and the worms are expelled. However, if you do the experiment at ZT12, then you're now favouring the development of a type 1 response, I beg your pardon, associated with IL-12, 23, 27 signalling. You get a type 1 signature and delayed expulsion. If you take the clock out of the dendritic cell, you then are forced down the type 2 response. You don't get an upregulation of the type 1 response. The type 1 response is you've lost that, what we call circadian gating. And that's important. So this idea that circadian time of day makes a difference to the outcome of infection is really important. Very rarely do you see papers which report time of day of infection, and yet from this data I think it has a massive effect on what then goes on in terms of the quality of the immune response, and subsequently whether your host is going to resist or succumb to a chronic infection. It's also important if we think about it in terms of vaccine strategies. So wouldn't it be marvellous if you could make your vaccine more effective just by giving it at a different time of day? And indeed, there's a lot of interest in this at Birmingham University, they published a paper a year and a half ago now, looking at flu vaccine, and they showed that it's 4 times more effective if you have your flu vaccine in the morning than in the evening. So those of you who have flu vaccines during seasonal flu, see if you can get it in the morning, not the evening. I think it also, and for this audience, it may be something to think about, I think it's also very important in terms of drug delivery. There's a huge interest in Europe on chemotherapy and how that relates to chronotherapy. So maybe delivering drugs at different times of day may make those drugs more effective. So it's 22. Am I okay for another 5 minutes? Yep. So finally, I just want to show you a few slides on the work which we're doing on B cells, because we think they are also important in determining this Th1, Th2 balance. So many of us think of B cells really as antibody factories. So B cells become plasma cells, and then they start churning out antibody, which is very important, and you can get antibody-dependent effector mechanisms. But of course, B cells are also really good antigen-presenting cells, and they act as B regulatory cells. So we've been trying to dissect out the importance of the B cell using a number of different model systems, the importance of the B cell in trichuris infection. And the experiments we've been doing involve depleting B cells in vivo. So quite simple, just using anti-CD20 treatment. And you can see that in our black 6 mouse, if we deplete B cells, we can make our mice more susceptible to infection. This just shows you the success of that anti-CD20 treatment. So we've got our CD19 cells are shown here. These are our B cells. If you've taken your B cells out with CD20, you've lost your B cell responses. Interestingly, this is genetic, it's strain dependent. So if you do the same experiment on a bulb C background, then B cells appear to play no role, and they're still able to expel the parasite in the absence of B cells. Perhaps unsurprisingly, B cell depleted mice make no antibody, but it's always very comforting to have that data there as well. So this just shows you parasite-specific G1, G2A and IgM responses, and there's nothing there if we've depleted our B cells. But more interestingly, B cell treatment has reduced our type 2 responses. So we've got a loss of 5, interleukin 5, interleukin 9, interleukin 13, and interleukin 10. Of course, that might just mean that we haven't got any antibody, and antibody is important in mediating the effector response. So what we then did was take away gamma interferon in our B cell depleted mice. So this is similar data to we've just looked at. Here's our isotype control. These are our B cell depleted mice, and you can see in the absence of B cells, mice are significantly more susceptible to infection. But if you now remove gamma interferon, so you're sort of giving those mice permission to mount a type 2 response, you've now got rid of your worms. So that suggests to us that B cells, the antibody isn't important in whatever those B cells are doing, and the B cell is acting much more as a regulatory cell and not as the step before lots of antibody, parasite-specific antibody production. We asked the same sorts of questions in a novel transgenic mouse, the IgMI mouse. So this was made by our collaborator, Ari Weissman. And so the details of how this mouse was made has been published in this paper quite a while ago, and it's not been a well-adopted model for some reason. Many people don't know it exists. It's a special mouse because this mouse has B cells, and it has B cell receptors of the IgM isotype, but it can't class switch that B cell receptor, and it can't secrete any antibodies at all. So it's a good way of retaining B cell, other functions of the B cell other than the antibody secreting capacity. So these IgMI mice are more susceptible to infection. And this is shown here at day 35 post-infection, and then again in a separate experiment through to 3 weeks post-infection. If you look at the cytokine responses, you see that the IgMI mice have reduced type 2 responses. So you can see that here 3 weeks post-infection and at day 35. So type 2 responses are down. But what's really interesting is that IL-10 responses are up. And actually they're up very, very early post-infection. This is day 10 post-infection. There aren't many cytokines that we can detect in the mesenteric lymph node just under 2 weeks post-infection. But IL-10 was expressed at really high levels. So when we did intracellular cytokine staining to see what the cellular source of IL-10 was, it turned out to be the B cell, which is shown here. So this is CD19 staining for B cells here. This is intracellular IL-10. And you can see that our wild types have a low number of B cells making IL-10, but this is upregulated to a significant level in our IgMI mice. This data just comfortingly shows IgMI mice make no parasite-specific antibody. So we think this IL-10 may be important in that susceptible phenotype. And that's really as far as I wanted to take it, but I think it's really important to think about B cells as regulatory cells as opposed to just antibody-producing cells. So we think if we deplete B cells, you lose support to get a type 2 response. We think on some genetic backgrounds, when a DC presents antigen to an unpolarized T cell, you need another cell. It's a 3-cell conversation in order to get to a type 2 response. On some genetic backgrounds, like the BALB-C, it's such a potent type 2 mouse that you don't need an accessory cell. But on mice like the BLAC6, which is making a mixed Th1, Th2 response, then the B cell helps to tip that balance towards Th2. And if you take that B cell away, you no longer can tip the balance towards Th2. Or if you use a mutant B cell, the IgMI B cell also no longer has the capacity to sort of act like an anti-gamma interferon antibody, if you like, and tip the balance towards a Th2. And thus, you end up with a long-term, persistent, chronic worm infection. Okay. So I will stop there, just a few acknowledgements. So Sarah Otto and Tom Hopwood are the 2 PhD students who've been involved with the circadian biology and the circadian rhythms, which has been a great adventure for me as a parasite immunologist, because I learned a whole new field, and I feel I've really learned a lot over the last few years. Rinal, Saputra and Emma, who's in the audience, has been involved with the B cell work. I should thank very much my circadian collaborators, Andrew, David and Julie, who've taught me a lot. David Bechtold is also a circadian biologist who helped us with all the telemetry work and made those fabulous clock physiology slides that I showed you early on in the talk. And everyone else in Andrew Loudon's lab and my lab. And the work is supported or has been supported by the BBSRC, MRC and GlaxoSmithKline. Okay, thank you very much.
Do we have any questions for Kathryn?
No, I don't need a microphone. My voice is loud. It's a bit of a weedy parasite. It is, I mean, you say it's chronic, but it's not really chronic. I mean, chronic parasites that I know of would be filariasis, for example. And there you have a really good example of periodicity of filariae coming out.
Yes.
So, I mean, presumably you're teeing up to do these experiments.
So I wouldn't regard whipworm as a weedy parasite. I think actually it's a really powerful parasite in terms of dissecting immune response, because it is actually truly chronic. It is a chronic infection in susceptible strains of mice. It's a chronic infection in humans. So humans will get low-level infections which are chronic and phenocopy our low-level infections in mouse models.
To convince me, I'd like to see it done in HP or polygyrus or something like that, something that is genuinely long-lived in the mouse.
So what would you describe as long-lived in the mouse?
Life of the mouse, basically.
Okay, well so if you look at longevity of the parasite, so we can track these parasites for 90 days and they're still there, and then I think they die of senescence. So
Yes, it's very interesting. Thank you. I was just, couldn't start thinking that biting behavior of mosquitoes and when they transmit certain parasites, and probably also influences the type of response, particularly in naive hosts. So malaria would be the first one where you get very, very strong Th1 response in more naive hosts, and it's actually not appropriate. It takes time to get to a Th2 in terms of a B-cell response, not necessarily a tissue response. So that's interesting to maybe
there's a
look
lot of
at experimental infections.
There are a lot of people, I think, interested in malaria, because malaria has a circadian clock that's not well worked out, but it is more understood than any sort of our parasites' clocks, because I'm sure parasites, these nematodes have clocks as well. I mean, mosquitoes bite when parasites are in the bloodstream, and
no, but also it's the mosquitoes bite mainly in the evening to infect.
Yes.
So that would, if that is translatable, then that would probably induce different, specific or more likely one type of immune response.
Yes, it'd be quite interesting to do malaria infection in clockless mice. No.
Thank you very much. Very interesting. A couple of questions. I'll declare my bias. I'm an animal scientist by trade, so I look at it that way. First question is, did you record feed water intake on the day versus night feeding mice and or body weight?
Mm-hmm.
And because I'm wondering if it's an indirect response that you're getting to some of those things, if it affected, for example, food intake.
Mm-hmm.
And this one may be a way off, but I realize that all the problems with finances and ethics and all the rest, but would the pig make a better model for a human than a mouse if you could do it? I mean, whipworms are a serious problem in agricultural. And I just wondered, the circadian rhythm of a pig compared to its metabolic body weight is much more similar to that of a human than a mouse.
Yes.
So the first one is about the feed intake, water intake, and the second is about the pig versus the mouse.
Yes. So they're both great questions. The answer to the first question is no, we didn't monitor body weight. So I can't answer that question directly for you. What do we know? I'm just trying to think. Yeah, no, I mean, we didn't do those experiments, so you can see that it's made no difference to the outputs.
But if you could record food intake on your groups of mice or whatever, daily food intake or something, it would be interesting to
Yes.
see the values you showed us
Yeah.
adjusted for food intake.
Yes, yes,
for
yes,
example,
yes,
Water
yeah,
intake or both.
yeah, yeah. No, it's a fair point, and no, we didn't look at that. In terms of pigs as a model, so I know Trichuris suis is a big issue in the veterinary world. I suppose the mouse model is a really tractable model, and we can look at mechanism really neatly in this mouse model, that I think mechanism would be really difficult to tackle in a pig. That's not to say that those experiments shouldn't be done as a stepping stone towards translation. Yeah.
So I think you've kind of answered my question with your slide, but with your activity levels being the same, does that mean they got very little sleep if they were day?
So we didn't do activity, so the telemetry, the whole body telemetry wasn't done in the reverse feeding experiment. So they were all just conventionally housed and fed ad libitum. So again, I can't answer. I don't know whether we'd get a change if we did that in the reverse feeding
I
experiment.
guess what I'm asking is the day feeding resulting in a lack of sleep, which has a much bigger effect on their immune response
Yeah,
than the
yeah.
feeding itself?
Yeah, so
If
we,
they're forced to stay awake and then they're just
Feed.
as active at night, then
Yeah.
they're getting no sleep. And I know what I'm like without any sleep. So it's just,
yeah, no,
you
no,
know, it's,
yeah.
That's just maybe like teasing that out in various experimental.
Yes, yes, so that could be looked at. If they're feeding during the day when they'd normally be asleep, then that might be affecting their longevity of sleep, but we didn't measure that either.
Also, with Britta's question, that was really interesting because I would have flipped it the other way and said that maybe mosquitoes bite when the immune response is least, and the evolution is faster on a mosquito than it is a human.
Yes.
Perhaps the response is more the mosquitoes than the humans.
Yeah, yeah, yeah. Yeah, I don't know. I mean, Sarah Rees is, do you know Sarah Rees? She's doing some stuff on this, isn't she, at Edinburgh University? And I don't know her story well enough.
So it'd just be interesting to see whether it is chicken or egg.
Yes, yes,
Because
yeah.
in terms of number of generations, obviously the mosquito kind of can adapt
Mm,
on a hugely easy basis.
yeah, yeah. Yeah, no, it would be really interesting to look at that, yeah.
Thank you.
Okay.
Okay. Well, it just leaves me to thank Kathryn Ells once again for a really fascinating talk.